Hi, I am Dr. Arsalan and today we are going to solve the MCQs on bacteria. First question is, dash is a pilus used by the bacteria for adhesion to the host cell. Given options are flagella, conjugation tube, fimbriae or hook. For example, if we consider this is a bacterial cell and there are some sort of projections arising from this cell wall of the bacterial cell, these projections are called fimbriae. Fimbriae are basically kind of pilus and are made up of pillin proteins, and these fimbriae are used by the bacterial cell to attach with the host cells. Option A is the flagella, which is used for locomotion. For example, there is a flagella in bacterial cell. This is used for locomotion. Conjugation tube is made between the two bacterial cells. This is made up of pilus. Conjugation tube is basically aimed for the sexual reproduction of the bacteria. The fimbriae, this is the correct option, that the fimbriae are kind of pilus made up of pillin protein. These are used by the bacteria for the attachment with the host cells. This hook is the structure present between the basal body and filamentous flagella. Next question is that dash is penicillin resistant bacteria. It means that the bacteria which is not affected by the penicillin antibiotic drug. Given options are Rhizobium, Staphylococcus, Streptococcus and Mycoplasma. So many are the bacteria present in the root of leguminous plants, while Staphylococcus and Streptococcus cause infections in the human beings. For example, throat infections, the respiratory tract infections, the pneumonia. These are caused by the Staphylococcus and Streptococcus bacteria. The last one is Mycoplasma. It also causes pneumonia, but here it should be kept in mind that the Mycoplasma, this is also called as atypical bacteria. Atypical bacteria means that it lacks cell wall. Plasma is the only bacteria which lacks cell wall, and this is the exceptional case of bacteria having no cell wall. And we know that the penicillin antibiotic, its mode of action is to reduce or diminish the bacterial cell wall. It basically degrades the bacterial cell wall, and the Mycoplasma lacks the cell wall. So therefore, penicillin antibiotic bears no effect on the Mycoplasma bacteria. So the option D is correct, that the Mycoplasma are atypical bacteria due to lack of cell wall is not affected by the penicillin. It's not sensitive to the penicillin antibiotic. Next question is the compost of organic matter which increases the soil fertility. Compost is basically made up of organic matter. Basically, when this organic matter is decomposed by the bacteria, the saprophytic bacteria, and the leftover is called compost, it is highly fertile and increases the soil fertility. So given options are detritus, humus, fertilizer, or both A and B. This compost, which is the degraded or decomposed part of the organic material or dead organisms, is also called as detritus or humus. So both options, detritus and humus, are correct. So we'll select the option D. Next question is, in bacteria, the basal body protrudes from the dash. Basal body is basically the structure from which the flagellum arises. In eukaryotic cell, the basal body is made up of the microtubules, and microtubules are the components of cytoskeleton. So in bacteria or prokaryotes, the cytoskeleton or microtubules are absent, and these basal bodies are protruding from the cell membrane of the bacteria, because in bacteria the cell membrane performs the function of the organelles. The given options are microtubules, cell wall, cell membrane, or flagella. That the basal bodies from which the flagellum arises is constituted by the cell membrane of the bacteria. Next question is, dash bacteria is present as symbiont in the roots of leguminous plants. The plants in which the symbiotic bacteria are present in the roots in the form of nodules, these plants are called leguminous plants, and these are rich in proteins because these symbiotic bacteria basically converts the nitrogen of the atmosphere into nitrates and then proteins. Bacteria are Bacillus anthracis, Rhizobium, Salmonella, or Azobacter. So out of these Bacillus anthracis and Salmonella, these are the bacteria which infect humans or animal population, and these are pathogenic. Azobacter is also present in the plants. The Rhizobium is the bacteria which is present in the nodules of roots of the plants called leguminous plants. These convert the atmospheric nitrogen into nitrates, which is further utilized for the protein synthesis in the plants. So the option B is correct. Rhizobium is the bacteria present in the leguminous plants. Next question is, capsule of Bacillus anthracis is made up of. Capsule is the outermost covering layer in the bacteria lying outside the cell wall. So its composition is asked here. The given options are polysaccharides, amino acids, cellulose, or murine. The polysaccharides, cellulose, or murine, these basically constitute the cell wall of bacteria, while the amino acids, these constitute or synthesize the capsule of the bacteria. So the option B is correct, amino acids. So the next question is, cluster of flagella on the poles of the bacteria is called. Given options are Lophotrichus, Amphitrichus, Peritrichus, or Atrichus. As we know that the flagella are present in the motile bacteria, and these may be present at the one side or both sides. Flagella may be present throughout the cell of the bacteria, or these may be absent. So the flagella, if present on one side, this is called Lophotrichus. If flagella are present on both sides, this is called Amphitrichus. If the flagella are present throughout the cell wall of the bacteria, these bacteria are called Peritrichus. If the bacteria lacks flagella, for example the cocci bacteria, these lack the flagella, these are called Atrichus. In this case, the cluster of flagella are present on the one pole of bacteria, so this is the correct option will be Lophotrichus. Diplococci bacteria are dash. Lophotrichus having flagella at one end, Amphitrachus having flagella on both sides, Peritrichus having flagella throughout the cell of the bacteria, or Atrichus having no flagella. So as we know that the cocci bacteria, these are non-motile, these lack flagella, whether these are the Cryptococci, Staphylococci or Streptococci. So the correct option would be Atrichus. Next question is, microbes were discovered by dash. Louis Pasteur, Antony van Leeuwenhoek, Frederick Loeffler or Dmitry Ivanovsky. Louis Pasteur, he developed the vaccines. Antony van Leeuwenhoek was the scientist who discovered bacteria for the first time and called these bacteria as the animalcules. So option B is correct. The Frederick Loeffler, he was German bacteriologist. He discovered various pathogenic strains of the bacteria, but he was not the first one who discovered the bacteria. The first scientist who discovered the bacteria was Antony van Leeuwenhoek, while Dmitry Ivanovsky, he discovered the viruses. Next question is, dash are the pocket-like structures or invasions of the bacteria for cell division. Given options are mesosomes, centrosome, lysosome or cell membrane. In case of bacteria, for example, it is a bacterial cell. This is cell membrane and outside there is cell wall in the bacteria. During cell division, this cell membrane of the bacteria invades inwardly and this form a pocket-like structure. This is called mesosome. So this mesosome deepens and this bacteria is divided into two by the invasion of this cell membrane called mesosome. So the option A is correct. While rest of the centrosome, lysosomes, these are the cytoplasmic organelles which are absent in the prokaryote cell bacteria, and cell membrane, it is basically invading. So this pocket-like structure invaded by the cell membrane into the cytoplasm of bacteria, this pocket-like structure is called mesosome. Next question is, gram-negative bacteria attains dash color by safranin dye. Given options are blue, green, red or violet. We should recall here that the bacteria can be divided into two groups, the gram-positive and gram-negative bacteria. This classification of the bacteria was brought forward by the Christian Hans Gram. He was the scientist who developed Gram stain, and the bacteria which are stained through this Gram stain and attain the color of blue or violet, these are called gram-positive bacteria. And the bacteria do not attain this violet or blue color and appear colorless microscopically, these are counter-stained by safranin stain, and this safranin stain give pink or red color to the bacteria. So these bacteria are called gram-negative bacteria because these were unable to absorb or capture the Gram stain, the blue or violet color, and are counter-stained by the safranin stain and appear red or pink. So the option C is correct, that these bacteria appear red or pink in the microscopic view. These were the MCQs related to the bacteria, its structure and importance. Thank you.